Hello, my name is Jessica Valadares, and I am very happy for the opportunity to show our study about the cytotoxic mechanisms of digitoxin monosaccharides analogs to modulation of cell oxidative stress and calcium homeostasis. Cancer is a leading cause of death worldwide, and its treatment is hampered by the lack of specificity and side effects of current drugs. In this context, 7 markers that allow tumor growth and spread have been reported. This work involved 2 of these markers: altered stress response and proliferative advantage that allows tumor progression. Thus, the discovery of drugs that act against these markers is an anti-tumor strategy. Tumor cells change calcium signaling to positively regulate uh, cell cycle progression, apoptosis escapes, and to increase mitochondrial energy production, promote the cell survival. This change is done through the modulation of the expression and activity of proteins involved in calcium homeostasis. However, calcium is involved in the regulation of many cellular processes, and increasing its concentration can induce cell death events. In addition, risk factors associated with cancer induce the production of small amounts of ROS, which induces the expression of proteins involved in the initial events and in tumor progression. However, the increasing of ROS can induce an oxidative damage to membranes, DNA, and proteins, and cell death, what can consist an anti-tumor strategy. In previous work, we demonstrated that digitoxin analogs were more cytotoxic and selective for healed tumor cells than non-tumor cells by inhibiting sodium-potassium TPase and plasma membrane calcium TPase in these cells, and they are potent inhibitors of purifying sodium pump. Thus, the first aim of this study was to investigate the anti-tumor mechanisms of these digitoxin analogs with respect to oxidative state in HeLa cells. For this, HeLa cells were treated with the analogs for 24 hours, and the levels of hydrogen peroxide, reduced glutathione, superoxide dismutase activity, and lipid peroxidation profile were evaluated. The results showed that they were established by glutathione reductase. The treatment with the analogs also reduced superoxide dismutase activity, probably by inducing oxidative damage in these enzymes and thus reducing its activity, which is important for the neutralization of superoxide anion. We observed that the treatment that increasing hydrogen peroxide levels also induces lipid peroxidation, which may alter the fluidity and integrity of the membrane, damage enzymes such as sodium pump and PMCA, which may impair the normal functioning of cells and inducing the cell death processes. In summary, the cytotoxic mechanisms of digitoxin analogs in HeLa cells involves the induction of oxidative stress by to decrease GS8 levels and superoxide dismutase activity in important antioxidants, and increase in hydrogen peroxide, which induces lipid peroxidation in the membrane. These effects were probably caused by inhibition of sodium potassium TPase and PMCA, which could lead to increasing intracellular calcium levels, mitochondrial overload, and hydrogen peroxide production. Thus, the analogs overcome the characteristic of adaptation to stress of tumor cells and inducing oxidative stress and decrease cell viability. The second object of this work was to evaluate the involvement of PMCA4 and important proteins for the calcium homeostasis in the anti-tumor mechanisms of the analogs. For this, we used HT29 cells because before reaching confluence, these cells have down regulation of PMCA4, which is used to promote cell proliferation. However, when they become differentiated, as within 5 days at confluence, these cells express a large amount of PMCA4 in plasma membrane. This allows us to assess the involvement of PMCA4 in the cytotoxic mechanisms of the analogs. Thus, HT29 cells before cell confluence and on the 5th day of cell confluence were treated with the compounds for 24 hours, and we evaluated cell viability and the expression of PMCA4, NCS1, MCU, and AIF in the two treatment conditions. The results show that in the condition of PMCA4 downregulation, the compounds were cytotoxic, but they lost this effect with the presence of this enzyme. This suggests that the cytotoxic effect of the analogs may be associated with PMCA4 down expression in the cells and the accumulation of intracellular calcium levels, which demonstrates that down expression of PMCA4 seems to have sensitized the cells to treatment with digitoxin analogs. And at the same time, it's also demonstrated that the cytotoxic effect of the compounds does not depend on the direct action in PMCA4. We also demonstrate that in the condition of PMCA downregulation, the analog rhamnose digitoxygenine increases NCS1, a protein that stimulates the release of calcium from the endoplasmic reticulum through the IP3 receptor, directing calcium to the mitochondria. This analog also increases the expression of apoptosis-induced factor, AIF, suggesting mitochondrial dysfunction and release of apoptosis factors. However, both analogs did not modulate the expression of the uniform mitochondrial calcium complex, MCU. In the condition with PMCA4 overexpression in the plasma membrane, the analogs did not modulate the expression of NCS1. They decreased AIF expression and seemed to increase the MCU levels, which is consistent with the absence of cytotoxic in this condition. In summary, we demonstrate that in the condition of PMCA4 downregulation, the compounds were cytotoxic. The compound rhamnose digitoxygenine increased NCS1 and AIF expression, which appears to be linked with intracellular calcium levels and mitochondrial dysfunction, leading to decrease the cell viability. However, the cytotoxic effect of the N-cetose digitoxygenine seems not associated with the modulation of these proteins. Thus, the downregulation of PMCA4 is used by HT29 cells to obtain a proliferative advantage. However, the analogs use these mechanisms to reduce the viability of the cells, probably by increasing intracellular calcium levels. In the presence of PMCA4 in the plasma membrane, allowing a greater control of the intracellular calcium levels, the analogs reduce AIF expression, which was consistent with the absence of cytotoxic effect in this condition. This suggests that the increase in intracellular calcium levels is important for the cytotoxic effect of these digitoxin analogs and highlight PMCA4 as an important target for the development of anti-tumor agents. I would like to thank Leandro, Dr. Giorgio Dorich, and Natalia for making this work possible, all friends of the Cell Biochemistry Lab, the university, and the financial support. So thank you.
Hi everyone, my name is Deise Carvalho. I'm from Federal University of Paraíba, UFPB in Brazil, and it's a pleasure to be here in this meeting. I will present my work entitled Molecular Docking and Antiviral Activity of Voabine against a Brazilian Zika virus strain. So, ouabain is a cardiotonin steroid that has several biological activities. Our group has been demonstrated this steroid as an immunomodulatory substance. We observe the ability of ouabain in reduction the neutrophil migration in several models, the pro-inflammatory cytokines also in several models, inflammation triggered by leishmania, the adhesion molecule CD18 and the p38 in neutrophils, and the away inflammation, demonstrating this way the anti-inflammatory activity of ouabain. Other works has been demonstrated the antiviral activity of this substance against a broad spectrum of viruses, such as HIV, influenza virus, and also the SARS-CoV-2, the virus that causes COVID-19. The Zika virus is an emerging arbovirus associated with neurological disorders, such as microcephaly, and at the moment, no specific vaccine or antiviral are available to treat this infection. So, in this way, the aim of this work was to evaluate the antiviral activity of ouabain against a Brazilian Zika virus strain. First, to do that, viral cells were treated with different concentrations of ouabain, and the cytotoxic activity of the substance was evaluated by the MTT-assay. We found the CC 50%, the concentration that reduced 50% of the viability of the cells, and the CC 20, the concentration that reduced 20% of the viability of the cells. And we consider this the maximum non-toxic concentration to the cells to be used in the antiviral assays. So, to analyze the antiviral activity of ouabain, viral cells were treated with the adhesion molecule CD18 and the p38 in neutrophils, and the away inflammation, demonstrating this way the anti-inflammatory activity of ouabain. Other works has been demonstrated the antiviral activity of this substance against a broad spectrum of viruses, such as HIV, influenza virus, and also the SARS-CoV-2, the virus that causes COVID-19. The Zika virus is an emerging arbovirus associated with neurological disorders, such as microcephaly, and at the moment, no specific vaccine or antiviral are available to treat this infection. So, in this way, the aim of this work was to evaluate the antiviral activity of ouabain against a Brazilian Zika virus strain. First, to do that, viral cells were treated with different concentrations of ouabain, and the cytotoxic activity of the substance was evaluated by the MTT-assay. We found the CC 50%, the concentration that reduced 50% of the viability of the cells, and the CC 20, the concentration that reduced 20% of the viability of the cells. And we consider this the maximum non-toxic concentration to the cells to be used in the antiviral assays. So, to analyze the antiviral activity of ouabain, viral cells were treated with this substance before and after the infection, and the antiviral effect was evaluated by TCID-50 assay and PCR. As we can see, here we have the virus titer in function of the different groups. And we see the ouabain has the ability to reduce the virus titer when compared to the control in the pre-treatment and also in the post-treatment, mainly in this assay. Also, ouabain was capable to reduce the RNA copy numbers of Zika in all concentrations when we compare to the control, but only in the post-infection assay. After that, virus cells were submitted to time of addition experiments with different times and temperature to analyze if OABINE has the ability to interfere in the entry into the virus into the cells. And the result was evaluated by TCID 50C. As we can see, here we have the virus titer in functions of the different groups. OABINE was not capable to interfere with the viral inactivation, viral adsorption, or viral internalization. This data suggests that the substance may not interfere with the entry, but can possibly interfere with the post-entry stage of the Zika virus life cycle. Finally, we perform in silico experiments to analyze if OABINE has affinity to non-structured Zika virus proteins. To do that, we perform molecular docking and molecular dynamics. So we choose three main non-structured Zika virus proteins, the NS3 helicase, the NS5 methyltransferase, the NS5 RNA-dependent RNA polymerase, or ADRP. As we can see, we found a total of 10 bind sites in the NS3 helicase, 2 bind sites in the NS5 methyltransferase, and 2 bind sites in the NS5 RGRP. After the identification of the bind sites, we analyze the delta gene of bind, or the energy free of bind, to the interaction between OABINE and the targets. The more negative the delta G, more affinity is the interaction. So we found that OABINE has a high affinity to the bind site 5 of the NS3 helicase and the bind site 1 of the NS5 RGRP, with delta G of bind of the negative 9.84 and the bind site 1 negative 10.58. This data suggests that OABINE has the ability to inhibition these proteins, and this could be related to mechanism of action of OABINE. Taken together, these data demonstrate the antiviral activity of OABINE against the Zika virus strain. Also, we demonstrate the OABINE could have the ability to interfere with the post-entry stage of the Zika life cycle, and the in silico data reveal new insights about the OABINE targets. So this work confirmed the effectiveness of the cardiotonic steroids as antiviral agents. Now we are working on to analyze the anti-inflammatory activity of the substance in neuronal cell line treated by Zika virus. In addition, considering that Zika virus infection causes intense inflammatory response in the brain, and OABINE has anti-inflammatory and antiviral activity, a question arises. Could endogenous OABINE be able to inhibit or modulate Zika virus infection in humans? So further studies are needed to better understand the role of the endogenous OABINE in these in humans, in disinfection humans. So I would like to thank the Federal University of Paraíba for the support, my group, the Immunobiotechnology Lab, advised by Professor Sandra Mascarenhas, in João Pessoa, the Oswaldo Cruz Foundation, also in Brazil, for the collaboration with the Virology Lab, advised by Professor Linda Lapena, and the collaboration with the Bioinformatics Elton for the molecular in silico data and the financial support. Thank you for your attention, and I'm available if you have any questions. Thank you.
Thank you, Daisy, for a nice presentation. So we now have some questions. Let me see. About the Zika virus effect, Leandro is asking. Do you believe that the modulation of the sodium potassium TPase by woabine could also be involved in the mechanism of action, or only direct effect on the virus protein?
Hi, everyone. It's a pleasure to be here. And thank you for the question. Yes, we believe that the sodium pumping involvement, and actually there are article in with the Chinese group that he actually did with Zika virus the modulation of oabine with the pump, and they see the inhibition of the pump in the Zika virus. So for do that, we performed the molecular dynamics to see the direct effect on the virus. But the group of the Chinese already did it in the last year this experiment.
Okay. Second question from Francois. Do you think that woabine will be able to attain sufficiently high concentrations into the cell contaminated with the Zika virus to inhibit the non-structural enzymes of the virus?
Just a moment. Yes, we believe that it's possible that oabine can be inside of the cell and do the direct action in the viral protein. But we have to do the experiment in vitro assay to know if there is possible. But we believe that, and we are working on to do this experiment in the future. Thank you for the question.
So I have a question about these Vero cells. Vero cells are kidney cells, isn't it?
Yes, that's
and
right.
the preference of Zika virus is to, I guess, neuronal cells.
Yes, but the viral cells is a lineage that is widely used for a lot of virus. So we choose to do the experiment in the cells first. And now we are working on to do in the neuronal cell line to analyze better the relation with Zika virus.
Okay, we don't have more questions. Thank you, Daisy, for the talk.
Thank you.
And now we
I appreciate
go on.
it.
Thank you. We go on with the next talk.
First, I would like to thank you, the organizer, for being selected for this talk. I am Thalita Maslow, and I will present to you the work I have developed at Federal Fluminense University in the Laboratory of Tissue Culture Herta Maia during my PhD with Elizabeth's supervision. In our work, we evaluated the effect of ouabain on retinal neuroinflammation and on retinal ganglion cell survival. Ouabain is a ligand of the sodium pump and at high concentrations blocks the pump effect and promotes a positive ionotropic effect on heart muscle cells, for example. For other hand, at nanomolar concentrations similar to circulating ouabain concentrations, this molecule can trigger several signaling pathways that influence gene expressions, leading cell growth, survival, proliferation, and immune regulation. Our group has studied the effect of potential molecules and pathways activated by ouabain on neuroprotection. Our experimental model is the retina. The retina is located in the posterior region of the eyes and is composed by neurons and glial cells such as microglia and Miller cells. Ganglion cells are neurons of the retina whose axons form the optic nerve. After injury, the optic nerve degenerates, leading to ganglion cells death. We have shown that when ganglion cells are axontomized, 50% of the cells die in culture, and the treatment of 3 with 3 nanomolar of the ouabain increase the ganglion cell survival. Because of this, we used the ouabain in 3 nanomolar concentration in all experiments of this work. Also, it's important to emphasize that almost all the sodium-potassium ATPase isoforms are present on the retina. Both alpha-1, alpha-2, and alpha-3 are present in different stages of the retina development. In the neonatal retina, we can observe a strong staining for alpha-1 and alpha-3 in the inner plexiform layer and ganglion cells layer. However, in the adult retina, exhibits a diffuse labeling for all isoforms. In the previous works, it was shown that ouabain effect on ganglion cell survival is dependent on NF-kappa B, a transcription factor important for inflammatory response. We also demonstrated that TNF-alpha is a critical cytokine for the neuroprotection mediated by ouabain on ganglion cells. In addition, ouabain induces the release of TNF-alpha mainly 24 hours after treatment. Based on this data, the aim of this work was to analyze the levels of important inflammatory receptors in the retina, along with microglial markers and ROS production after ouabain treatment. Also, we tried to understand by which mechanism ouabain increases ganglion cell survival. We performed cultures using the retina neonatal rats. This tissue was desiccated, dissociated, and plated in petri dishes, and then the cultures are treated with ouabain by different periods. These cultures were incubated by 37 degrees and used to Western blot, acridina, orange assay, and rose assay. To survive experiments, we did a peroxidase injection at superior colliculus, and 16 hours later, we performed retinal cultures. The ganglion cell survival was analyzed at 48 hours. In this image, we can see the ganglion cell staining with peroxidases. So we analyzed some of the main inflammation markers in the retina after the injury, such as TNF receptor 1 and 2, Toll-like receptor 4, CD14, and ABL1. And we observed that the levels of almost all were downregulated by OA-BAM treatment, except for TNF receptor 2, which was increased after 24 hours. We also observed that OA-BAM was able to reducing reactive oxygen species shortly after injury. Moreover, we investigate the apoptosis pathway in the OA-BAM effect. And using apoptosis inhibitor, we observed that is not alter the OA-BAM effect on survival. And also, OA-BAM treatment not change the levels of proteins involved in the apoptosis, such as phosphorylated JNK and caspase 3. In addition to this, we observed that OA-BAM increased the expression of the proteins involved in autophagy, as BCLIN1 and LC3. So we ask you whether autophagy could be a potential mechanism for retinal ganglion cell survival induced by OA-BAM. And using an autophagy inhibitor, we observed that the neuroprotection of the ganglion cells mediated by OA-BAM depends on autophagy activation, which was corroborated by OA-BAM leading to an increase in the formation of autophagoson. Together, these results show that OA-BAM increases the ganglion cell survival by an autophagy-dependent mechanism and also regulates neuroinflammation and ROS production in retinal cells cultures. This is our group at Federal Fluminense University that contributed to this work. And also I would like to thank our collaborator at UF, Unirio, Fiocruz, and UFRJ, and the supervisor Elizabeth Gestal de Araújo. Thank you for your attention.
Thank you, Thalita. Great talk, great work.
Thank you.
Let's see if we have questions. So I have one question. So you were working with OA-BAM for a long time. Your lab is working with OA-BAM for a long time in retina. Do you think that other cardiotonic steroids have the same effect as OA-BAM in the case of survival?
The other cardiotonics?
Yes, other cardiotonic steroids like digoxin or like bufadienolide and so on.
I believe so. Could be an interesting question. Maybe we can evaluate it, but I don't know. I believe yes, but I don't know.
And what do you think? What do you think is the meaning of protection of OA-BAM in this kind of cells? Do you have any clue why it's protecting? Why so low concentrations of OA-BAM is protecting? Maybe you have some endogenous OA-BAM in this system, so it works like that.
Wait a minute, please. About the signaling, the effect of the OA-BAM. Can you repeat, please? Because cortou pouco.
Não, aí depois eu posso responder. Por que você acha que... Qual a razão da OA-BAM fazer isso nesse tipo de célula especificamente? Por que você acha que concentrações tão baixas de uobaína protegem essas células? Existe alguma razão que você possa vislumbrar?
The uobain can neuromodulation of this tissue. When we dissect this retina, made a culture, you automatically induces a lesion on the cells. And when we use your uobaina treatment, we regulate the production of cytokines like TNF, alpha, and EL1, 2, 6, and some factors like proteins pathways important to survival of the cells. The uobain controlling the inflammation homeostasis in the system.
Thank you. There's another question from Dr. Carlos Frederico Fontes. Are these effects blocked by PP1 treatment?
I don't know. Maybe it's a important pathway to analyze. That the block is dependent on PKC, protein kinase C, independent of the calcium, JNK, NF-kappa B, but is independent on independent on I forgot now. Independent sorry. But I know that dependent the pathways, but PP1 I don't know.
And one more question from Dr. Gustavo Blanco. Is the effect of uobain on retina only on neuronal cells, or it also extends to the photoreceptors?
Okay, thanks. In our model, the retina is immature, and some cells are differentiated, others not yet. Photoreceptor, some photoreceptor is present, is differentiating this tissue. But on photoreceptor specifically, we don't know. But certainly the uobain acts in other cells, in other neuronal cells, and in glial cells, like microglia.
One last question. Do you think uobain can challenge the system by activated NF-kappa B?
I think so, because this factor is important to in the production of cytokines, and we could see that uobain regulates all cytokines and this factor too. So yes.
Okay. Thank you very much, Talita, for your talk. It's a beautiful
Thanks.
work. And then we go to the next one in minutes.
So, hello everyone. Hello everyone. In 10 minutes. Hi everyone. Could I share my work now? We can see. Okay, sounds great. Okay, I will start sharing my screen. Like here. Okay. Hello everyone, my name is Jue Zhang. I am a postdoc fellow from Dr. Yiliang Chen's lab at Varsity in the US. And actually, I also had a training and learned a lot from Dr. Zijian Xie and Dr. Senjun Pierre when I was at Marshall University. And today, I would like to share this topic. Sodium potassium ATPase is a negative regulator of LPS-induced macrophage activation. First, I will introduce this background briefly. As we know, macrophage is one type of innate immune cells. It could help our body to eliminate the microbes. And the lipopolysaccharides, this LPS, is a major component of gram-negative bacteria. And LPS is known to bind with different Toll-like receptor members, and especially this TOL4 receptor, and promote the downstream signaling pathway, including NF-kappa B pathway, and promote the pro-inflammatory cytokines production. But however, the excessive pro-inflammatory response might induce some pathological disease, such as sepsis. Therefore, the negative regulation and the balance is important. And we all know this NKA is an L transporter as well as a signaling transducer. And based on Yiliang's previous study, he found that NKA is also involved in macrophage inflammatory response. Because these cardiotoxic sterols, WABN, could bind with NKA and CD36 complex and promote the downstream signaling pathway, NF-kappa B pathway, and eventually promote inflammation. However, the role of NKA in LPS-triggered inflammatory response and the detailed mechanism is still largely unknown. Therefore, the aim of our study is to explore the role of NKA signaling in LPS-induced inflammation with our NKA haplodeficient cell model and mice model. So here is the data. First, we isolated peritoneal macrophage from the Y-type and NKA heterozygous mice, and then we treated these cells with LPS 100 nanogram per milliliter for different time. So first we check the mRNA level of these different pro-inflammatory cytokines. So here we could see LPS increase these cytokines RNA level in NKA deficient cells. And next, we double check the protein expression level of these cytokines from the post-culture medium. Again, we see this LPS increase these cytokines protein level in NKA deficient cells. So this data could suggest NKA haplodeficiency enhanced pro-inflammatory response induced by LPS. So next, we performed some in vivo study. We IP injected LPS into Y-type and NKA heterozygous mice, and then we collect the blood plasma before and after LPS injection and compare the cytokines level in this plasma. So here, figure A shows this data. Again, we see this cytokines level increased in this NKA heterozygous mice after LPS injection, which is consistent with the in vitro study. And we also observed their mice survival for 7 days. So this is the survival rate data. Here, this is NKA heterozygous mice group. We could see these NKA heterozygous mice are hypersensitive to this LPS-induced lethality. So all this data suggests NKA heterozygous mice are hypersensitive to this septic shock. And next, we explore more mechanisms through this NKA Src family kinase and TOL4 complex signaling. We performed co-IP experiments. First, I isolated these TOL4 precipitates from cell lysates and probe these precipitates with NK alpha 1 antibody and phosphorylated link antibody. So here we could see LPS actually decrease this NK alpha 1, this assembled NK alpha 1 in Top4 complex, and also increase this phosphorylated link in this complex. And next, we compare this co-IP data in Y-type and NKA heterozygous, and NKA deficient cell group. And we can see when NKA is deficient, it increases more assembly P-link in this Top4 complex. So this data, we could conclude NKA negatively regulates this Top4 link complex induced by LPS. And recently, I am performing the reverse co-IP experiments to confirm this interaction. And also, we are trying to do the PRA experiment to confirm their in situ protein-protein interaction. So next, we explore more mechanisms through this NF-kappa B signaling pathway. And figure A showed this bunch RNA sequence data. We could see LPS could upregulate these NF-kappa B target genes. And this GC analysis could help us to confirm there is a significant difference between these two different groups. And next, we check this protein expression level of phosphorylated IKK. We see the kinase could promote this I-kappa B alpha degradation. Therefore, it could promote the NF-kappa B activation. So here we see LPS prolonged this IKK activation and also prolonged this I-kappa B alpha degradation in these NK deficient cells compared with this Y-type group. And we double check this nuclear NF-kappa B expression level, and the nuclear NF-kappa B also increased in these NKA deficient cells. So this is a Western blot data. Furthermore, we confirm this NF-kappa B nuclear translocation with immunostaining experiments. So here, this green color indicates for NF-kappa B signaling, and this blue one indicates for the nuclear. So majorly we will check this merge image. So here in this NKA deficient cells group, we could see LPS increase this NF-kappa B nuclear translocation. We could, and there is less NF-kappa B located in this cytosol when we compare with this Y-type LPS group. So all of this data suggests NK deficient could promote more NF-kappa B nuclear translocation. Last, we also check the NF-kappa B transcriptional activity with a commercially available NF-kappa B reporter assay. And when NK look down, this black bar, this black group, when NK look down, LPS also increase this NF-kappa B transcriptional activity. So all of this data from figure A to figure E, it could indicate that NKA haploid deficiency increases NF-kappa B activation induced by LPS. And the last, we further check the metabolic change, because this metabolic change could also contribute to the pro-inflammatory response. So figure A showed the bunch RNA sequence data. LPS also increased upregulated this glycolysis-related gene expression. And we also conduct this seahorse assay with this ECA, which indicate for the actual cellular acidification, and OCA indicate for this cell respiration. And this blue line indicate for this NKA deficient group. We could see LPS increase this glycolysis rate and inhibit this cell respiration in this NKA deficient group compared with Y-type and LPS group. So this data could further suggest that NK deficiency could actually lead to a metabolic switch from the oxidative phosphorylation to the glycolysis. So here we conclude that NKA is a negative regulator of LPS-induced immune response by regulating this Top4 NKA link complex and the downstream NF-kappa B signaling pathway in macrophages. So thank you. This is my presentation today, and this is a picture of our lovely lab members. This is Yi Liang, this is me, and our lab technician Yi Qiong and Jackie. And we also have a lot of collaborations, including also we have collaboration with Dr. Shapiro and Dr. Sodi at Marshall University. So thanks for your listening. Please let me know. Oh, sorry. Please let me know if you have more questions.
Thank you very much, Zhu. It's very, very interesting and deep work. A lot of important results. Let me see, we have questions. I have one question for you about this inflammatory role of sodium potassium TPAs, because there are some works that show that wobain, that's the ligand of sodium potassium TPAs, would have an anti-inflammatory effect.
Yes.
So how you see this with the results that you show there? And do you plan to use wobain in your system?
Yes, thanks, Louise. Good question. Actually, there are several papers already published. When I went back to the literature, we could see a lot of people use wobain to treat different cells like cardiomyocytes or other or kidney cells, and they found that wobain could inhibit the NKA pumping function. But if they could protect from, for example, like the sepsis-related shock or not, there are some debates there. That's why in our study we want to focus on this NKA knockdown or NKA deficient model to try to explain more about the mechanism. And you're right, a lot of people already focus on this wobain effect. So in our future study, we won't repeat this again. Thank you.
Thank you. From Sandra, Dr. Sandra, great talk. Have you observed MAP kinases involvement?
Yeah, thank you, Sandra. Also a good question. You are right. We check the MARC, ERK, and the PS3K-AKT signaling. But we didn't present here because in these NK deficient cells after LPA treatment, we didn't see much significant change about this MARC signaling pathway. But we only observe this PS3K signaling increased, lead to more increment in these NK deficient cells compared with wild type LPS group. So that's why, but only PS3K signaling changed. That's why in our next paper, we may just present this data in our supplemental documents. But here we'll still focus on NF-kappa B signaling. Thank you.
Thank you. There's another question here. Nice talk, Zhu. Is the animal model in this animal model? Do you know if alpha 2 is more active to try to compensate alpha 1 decreased activation?
Oh, good question. You focus on a different isoform. First of all, in this alpha 1 lookout model, we checked alpha 2 express